All right. So hello, everyone. Welcome to BioTuring Webinar. So today we have a special guest, Matan Hofree, which I haven't seen for the last, I believe, 10 years, more than 10 years. And he will talk about the tissue level insights from cellular measurements, identifying multicellular hubs in colorectal cancer. And about Matan received his Bachelor in Computer Science and Biology from Hebrew University of Jerusalem. And about 15 years ago, we shared some path as Matan did his PhD in computer science at UC San Diego, working with Trey Ediker, where he developed the clustering, classification, inference approaches using prior biological knowledge encoded in gene-gene interaction networks. And then after that, I believe that he went to the Broad Institute to work with Aviv Regev, and now he's currently a scientist at the Weizmann Institute. And Matan develops computational analysis technique for studying cancer and complex genetic diseases using algorithms and ideas from machine learning and graph theory. So he focuses on advancing our understanding of interplay between genetics mutation, transcriptional changes, and cell-cell interactions, and how these drive the emergence and evolutions of tumor. So it is my great pleasure to introduce Matan. And Matan, I'm actually looking forward to the talk and the work that you did with, I believe, Karin Pelka and Nia Hakohen and Aviv Regev, and of course many other colleagues. And more than that, I'm looking forward to hearing about the progress, your progress in the last 10 years that we haven't. All right, thank you so much, and I leave the stage to Matan.
Thank you so much for that very kind introduction. Let me make sure my screen is shared. Right, can you see my slides? Yep.
Yes.
Okay, and let's dive in because I have probably a few slides too many. So thank you for that very kind introduction. As Sean mentioned, we go way back, and we really need to catch up over a beer sometime soon, hopefully in person. Today I'll be presenting on the transcriptional landscape of colon cancer and how we used single cell analysis to identify multicellular hubs. So really the past 2 years, a little longer, where I've been focusing most of my efforts on colon cancer. Colon cancer is an important and impactful cancer type to study. It's the second most common cancer type overall when you examine both males and females. And it exists in 2 distinct subtypes, which is one of the things that really attracted us to study this disease using single cell and genomics approaches. One archetype is the mismatch repair deficient, or MSI high. It's characterized by microsatellite instability, hence the MSI high name, but it is actually due to mismatch repair deficiency. This is actually the less frequent subtype. The more frequent subtype is the mismatch repair proficient, or MSS, which is characterized by a lower tumor mutational burden, right? Basically, this is a slide from TCGA, just showing you the this almost this gap in the mutational frequency scale. And one thing that's interesting about these 2 cancer types is that the mismatch repair deficient is very immune active, sometimes referred to as immune hot, as opposed to the mismatch repair proficient, which is immunologically cold. And the reason that is interesting is we today have really realized that immune cells play a key role in controlling and preventing cancer in the healthy host. And they can also be weaponized. They can also be used, brought to bear to treat tumors, right? So one of the very promising approaches is by immune checkpoint inhibitor therapy, right? Anti-PD-1 is a very familiar type of therapy where we basically reactivate the immune system to attack tumors. In particular, in the sorry, I have some mouse issue here. Right, so in CRC tumors, right, these two archetypes behave very differently in terms of their response to immune checkpoint blockade or immune therapy in general. Right, the mismatch repair deficient, right, are clinically treatable by these therapy types, whereas the mismatch repair proficient are unresponsive to these types of therapies. And really, we wanted to understand the difference between these two diseases that occur within the same tissue in hopes of basically understanding, why only 50% respond, right? And can we identify further opportunities to treat mismatch repair proficient patients? So we recently published, right? This was published in September of last year, our cell paper on spatially organized multicellular immune hubs. And I'll give you the brief rundown, right? So our goal was to understand how malignant immune and stromal cells interact to shape the immune responses in colorectal tumors. And to do that, we aggregated a cohort of mismatch repair deficient and proficient tumors from multiple hospitals in the Boston area. Boston gets enough of a traffic that we are able to collect tumors in a manner that's fairly balanced between mismatch repair deficient and proficient tumors. And we basically characterize these using single-cell RNA-seq, taking care to capture multiple aspects of the tumors I will describe in a couple of minutes, and also adjacent normal samples from more than half of our patients. So this is was a massive effort, right? So it spanned in total over 4 years since the beginning of collection until publication, right? Over 500,000 cells, 62 patients, 100 specimens, 60 billion reads that were aligned, 5 terabytes of data. So really massive effort that was really made possible using the cloud computing resources, right? In particular, Terra Firecloud. I know some of these things are now easier to do, but at the time they were quite a challenge. And I know that Sean has a lot of ideas on how to make these things more easy. But when we started this, we didn't have such solutions available, right? And all of this was quite a challenge that we're not going to talk about all today. I could give probably a separate talk on that topic if there's sufficient interest. So basically serving, right, we used single-cell RNA-seq, right, characterizing both unsorted tumors and immune-enriched samples using 10x. And we created this atlas spanning, at the end, after all of the QC, 370 or so thousand cells from both the CRCs and adjacent normal. And we applied, one of the classic, I developed my own version of this, but it follows the general principles of using hierarchical graph clustering. And it really could sort of see these, the basic cell types really breaking apart very, very clearly, right? Epithelial cells breaking apart from the other immune types. And we applied a hierarchical approach to sort of identify a total of 88 different discrete clusters within this cohort. So I'm going to show a bunch of t-SNEs in case somebody has landed here and somehow doesn't know what a t-SNE is, right, or UMAP, right? It's just a technique for visualizing very high-dimensional data in a two-dimensional visualization, right? And I think it's really key to understand that it's a visualization technique, right? It's a communication tool, right? I don't do any sort of calculation on this visualization, basically, right? Everything happens using different approaches, but it's very useful to promote a healthy discussion of what's going on. So for example, showing here on the left, again, like colored by cell types, right? On the right, colored by the different types of specimens, right? So you can really see how the region that is, can you see my mouse when I'm pointing this way? Yes, no?
No.
No. Oh, I need to point my mouse this way for it to happen. Now you see my mouse.
Now it's your mouse.
Okay. So I'll do my pointing in the right place. Moving forward, right? So here you can sort of see where there's a distinct coloration, right? That's because we have the tumor part, right? Here where there's mixing, you sort of see the normal epithelium, right? And you see how there's very nice mixing between the different cell types when we examine, right, the different immune compartments. And one of the things that's really, I think, the power of single cell analysis techniques is you can zoom into each one of these compartments and further interrogate it, right? So here I'm just showing you an overview of the immune landscape. And really a lot of the things that we expected to see emerge in this view, right? We see an enrichment for T cells and myeloid cells. And again, like because it's sort of, you sample some pool, we see a depletion, right, of other immune populations. Zooming in a little further, right, I wanted to highlight a couple of the differences that were really apparent, right? We sort of see an enrichment of Tregs and monocytes and macrophages, right? You could sort of see Tregs in particular here, right, and monocytes in this red region in the middle. And you can really see how these populations really expand within our tumor cohort. When we compare to the normal populations that sort of show an enrichment, right? It's more of a depletion within tumors of B cells, right? Plasma cells and some populations of T cells. Zooming in further, right, into the T cells, which was to us one of the more interesting compartments, right? We could sort of characterize particular populations that emerge within the mismatch repair proficient, for example, these IL-17 positive, CD4 positive, and CD8 positive T cells, or within the mismatch repair deficient, we identified an enrichment in populations of CXCL13 positive and activated gamma delta-like T cells. The CXCL13 positive cells, if somebody is familiar with the biology of tumors, right, in the tumor immunology, in the literature, they're mostly reported as being part of tertiary lymphoid structure. So these are basically localized lymph-like structures that emerge around tumors, right? And the thinking in the field was that these CXCL13 are primarily localized within these structures, but we actually, when we went back and checked using microscopy, we could identify populations of these cells deep within the tumor tissue, which was quite a surprise. There's a lot more going on, really, and I can't go into depth on any aspect here. I just wanted to also show the dramatic expansion within the myeloid populations, right? Really, you sort of see dramatic enrichment in monocytes and macrophages, but also in granulocytes, right, and various types of dendritic cells, right? So there's really a lot going on in terms of the myeloid populations. Our cohort was actually not designed to capture well stromal cells, right? It's very difficult in one single cell experiment to capture all types of cells due to the differences in adherence, right? There's sort of a disassociation step where we break apart the tumor tissue. And if you want to keep your epithelium intact, you basically lose your stromal cells that are very, very sticky, right, and hard to disassociate. But even so, we are able to capture about 5% stromal cells, and we see very dramatic remodeling that we can report on in terms of different types of stromal populations, right? So we basically see depletions in certain fibroblast populations and the emergence of distinct cancer-associated fibroblasts, right? That appear to activate transcriptionally different aspects of the stem cell niches and the differentiation niches that we usually observe within the healthy dividing gut, right? So basically, these are really instrumental in the maturations of cell within the healthy gut, and we sort of see their activation, right, and enrichment within particular populations of cancer-associated fibroblasts. Zooming into the epithelium, which is really, I think, like everything, like there's challenges in every direction here, but I think a unique challenge is understanding and analyzing tumor tissue using single cell techniques. One reason for this is that there's a lot of patient-specific structure that is very salient and dominates a lot of the signal. Here I'm just coloring that same overview TC that I was showing you before, and colored by the different specimens, right? And you could really see how there's these islands, right, that are basically tumor-specific islands, right? Because there's a lot of tumor-specific substructure, right? And this is after taking extra care and doing taking great efforts to sort of basically characterize things in an even fashion, right? To sort of see the mixing with an immune population as opposed to these patient-specific islands. And maybe this is not that surprising due to various different features, but at the lowest level, here I'm showing you a heat map right on the right, where you could sort of see the rows are genes, right, and the columns are individual patients, and you could really see how there's unique patient level expression, right, in each one of these samples, surprisingly, also within normal tissue, right? So this is tumor adjacent normal, and so I think there's something very interesting going on that front as well that we're further exploring in subsequent studies. So really, we one of our goals in this study was to sort of find the shared structure within this high dimensional measurement that we capture using single cell RNA-seq, right? And really the basic building block that has been the focus in many of these studies is identifying the different clusters, right? One can think about clusters as really the simplest way to reduce data into these discrete types. But really, if we sort of take an analogy from a completely different space, let's say a car engine, right? You could sort of describe things in terms of their similarity along different axes and dimensions, right? For example, right, you could sort of organize the parts of this engine according to their shape or size, right? So there could be many different ways in which it's interesting. So a slightly different analogy is that can be drawn here is from looking at colors, right? So for example, if you were to sort of think about the Crayola palette, right? You could sort of, historically, there were only a few colors, right? This plot on the right sort of shows you the evolution of Crayola colors that are being sold, right? But of course, when you think about color, right, even though professionally, right, we can, Pantone, right, sells over nearly 2,000 different colors that have a unique name in their catalog, right? One could really describe all of the colors, right, using a continuous three-dimensional description, right? And so this sort of analogy really translates also to many different domains and also into biology. And we want to sort of find a continuous description of this data, and we turn to an approach that I think has been used before for analysis of transcriptional data, and is actually quite scalable and powerful, which is non-negative matrix factorization, right? So due to the fact that it can be solved using relatively simple numerical approaches, it is highly scalable and fast. Also applies well to very sparse data. Again, due to the fact that we just have very efficient algorithm for solving linear algebra problems on sparse data. So this really, I think, wonderful paper from Li and Xiong from '99, which I very much recommend reading, also shows that it has certain features that I think are very conducive to biology, right? They sort of capture what they called part space decomposition, mostly due to the additivity of these solutions, right? And this tends to capture well, like in their paper, they show how it finds different parts of a face, right? But we could also show that it captures certain biological properties, right, coherently. One of the main drawbacks is that the solutions are not unique. Okay, so basically this is dependent on the initialization, right? It's the problem is not convex. It is convex if you decide to fix W or H, right? But then you're sort of stuck with initializing either W or H. And one could ask or wonder out loud, why not use, say PCA, right? What is the big deal, right? And so one of the things that when you examine high dimensional data, PCA tends to basically choose as eigenvectors, right, things that are intermediates, right, things that span the space optimally, and not things that are best characterizers of different parts of the data, right? So this is just an illustration of what's going on, but I think it really drives home this point that NMF has the property that it also is a characteristic of your actual data. And so we wanted to sort of overcome some of these drawbacks. Mostly we wanted to get more robust results and also, if possible, improve the biological relevance. And so I developed a consensus NMF approach to basically run NMF multiple times and then aggregate the results. I don't want to spend too much time on the details here, or we can go back to this, right? But the key idea, right, is running NMF many times. Using different initialization, I try to introduce more variability by subsampling both genes and cells. Then I remove outlier programs and cluster the NMF solutions. And finally, I basically use that aggregate solution to reinitialize an NMF procedure, right? Resulting in, again, optimal, less dependent on initialization output. Which I then finally choose the optimal K using variance explain type approach. So I have various slides that I sort of hid away, but I just want to show, you know, something very briefly about the robustness, right? So this is comparing, right, the CNMF solution, right, the unique solution we get from this procedure to what you get when you compare to many vanilla initialization, and you can really see how in terms of both the median, right, and the lower quartile correlations, right, it's actually a lot more coherent. So at the output, right, what we basically get is different types of programs. Some of them are really cell type specific. For example, there's a program that is unique to PDCs, a particular type of DC cells, right? And some are really shared, right? For example, a program that we find within myeloid cells, right, that we can identify in different types of cells, including epithelial cells. Right, and we could go into each one of these programs and rank the key genes that sort of form the basis for this NMF decomposition and identify programs that my biologist colleagues were really excited about, because they really seemed to form coherent sets that were well described in the literature. And while one thing that I sort of didn't maybe stress well enough is that we do this separately for each one of these different compartments to really create as a comprehensive a dictionary as possible of these different programs, right? And we end up with 204 NMF expression programs within each one of the different cell lineages, right? So here I'm just showing you the T cell ones, right? But it's really a massive resource of different de novo discovered programs, right, that we find in each one of these compartments. And we can dive into these, right? So here I'm showing you a heat map where the columns are different patients, right? And the rows are different programs we found within these epithelial cells. And really a couple of, you can really see how these split apart nearly perfectly, but not perfectly between the mismatch repair proficient and deficient. You sort of see a subset that aggregates across the aisle, basically. And we can zoom into certain programs that really seemed interesting to us because they involved the activation of particular immune related properties. For example, MHC, right, interferon stimulating genes, and all sorts of inflammatory chemokines, right? And we could really see how these are dramatically more active in mismatch repair deficient tumors, right? When we compare to TCGA, we really see how some of these trends are well captured even within bulk tumors. But I really want to understand some of these relationships between the different tumor types. Unfortunately, that is not trivial to do using single cell analysis, right? There's also the spatial techniques that one could use here, but they have their own sets of drawbacks. And really, when we started the study, they were not mature enough to do this at this sort of scale. And so I developed an approach which we called covariation consensus NMF, which is just a way of uncovering covariation structure between these different programs, right? So the key idea here is to summarize the cell type specific expression programs at the tumor level and examine correlation across the basically the distribution that we find within each tumor, and then use that to uncover, right, or to identify using an appropriate significance test, a subset or subnetworks of closely interacting cells by basically uncovering close, frequently co-varying expression programs across different cell types. And we applied this approach in both mismatch repair deficient and proficient tumors. And really, within our study, we only have so much space within one manuscript. So we chose to focus on the less studied and more novel mismatch repair deficient tumors. Here we were immediately drawn to two interesting subnetworks. One, we call it an inflammation driving subnetwork, which was characterized by basically recruitment of or activation of neutrophil programs, right, Treg programs, myeloid inflammation, right, and also this malignant inflammation that I was highlighting before. When we zoom in, right, or visualize, right, this is just showing you a different view of the same sort of data, just showing the statistically significant edges. We see what looks like very frequent and robust interaction between a particular epithelial program and these myeloid programs. And we again could sort of see how this is dramatically different when we compare between the mismatch repair proficient and deficient and the normal tissue, but it is actually quite similar between these two tumor types. Just for the sake of time, I'm going to skip to a different network that we also examined, which is looking at a multicellular immunity hub that really involved key fascinating players from the immune environment of these tumors, right? So we were again, really, we were most drawn to these basically hubs that showed both malignant epithelium and immune cells, right? So here we sort of see activation of MHC, an interferon stimulating gene, right? Activation of a program that's related to CXCL13 and 11, right? And then involves the recruitment, right, and proliferation of T cells. And this CXCL13 T cell program that I sort of mentioned before was actually quite unique to us to uncover within the depth of these tumors. So here I'm comparing how this subnetwork looks within the mismatch repair deficient and proficient, and you can really see how this coherent structure really breaks apart, right? So what I'm showing here is in these dashed line, right, these non-significant edges, and you can really see how many of the edges are quite significant when we look at the mismatch repair deficient, right, but lose their significance within the mismatch repair proficient tumors. And again, this core activation of these programs is more active within mismatch repair deficient, but it is still enriched in mismatch repair proficient tumors as compared to normals. Again, drawing your attention to the fact that we saw, or we suspected, given some of this initial microscopy, that it might actually be an integral part of the response of these T cells within these tumors, right? Leading us to hypothesize that basically there's due to interferon gamma from these activated T cells that induces interferon stimulating gene programs within the malignant and myeloid neighboring cells that in turn basically recruit or further recruit these CXCL13 T cells into the depth of these tumors, right? To sort of follow up or at least tentatively validate some of these results, we turn to RNA-scope, which is just a way to interrogate spatially using microscopy a selected set of markers, right? These markers were uniquely selected to characterize the key parts of this program. And I'm somewhat colorblind and really bad at reading these sorts of plots. So I asked one of my colleagues to carefully annotate it here. And you could really see these foci where we see in a certain region of the tumor right, activation of both the CXCL10-11, right, the presence of CD3, right, which is showing us the presence of T cells, right, CXCL13, which we said is the marker for this unique T cell population, right, and the presence of interferon gamma that you see here in green, right. We sort of see these basically islands where all of these things come together deep within the tumor tissue. So just to show you, this is not totally cherry picked. Here I'm showing a smaller magnification, right? So you could basically see a wider field of view, right? And these things are basically these cities or hubs that occur deep within the tumor tissue. We of course do some further computational analysis, sort of show that this is not a one-off phenomenon, right? So we computationally characterize these regions by basically clustering and segmenting for these hubs within several multiple slides from different patients, and really see this correspondence, right, between the presence of cells within foci, right, and the activation of both the CXCL13 program or markers for the CXCL13 programs and markers for this epithelial interferon stimulating gene program that we find within the epithelial cells. So really, this sort of leads us to suspect that there's this crosstalk that involves interferon gamma and CXCL10 and 11 that basically induces the recruitment of these T cells into the depth of these tumors. And we also suspect that it is involved in the eventual exhaustion of these T cells populations, but also possibly in the formation of these tertiary lymphoid structures. So wrapping up some of the things that we sort of created as part of this study, right? So there's an expression program website that you could explore the 204 different populations, right? You can go into this companion website and basically explore the different programs, see what where they're active, which cell types they're active in, what genes they're characterized by, what are the particular gene sets that are these key genes are characterized by. So at the time, there were not many ways to sort of share data from single cell studies. So we use a single cell portal from the Broad Institute. And I found a nice way to sort of put all of our results within 10x loop files, which was actually not trivial, but they're also now available, I'm happy to say, from BioTuring's really phenomenally useful looking BB browser. That I think really my goal with this with these things that I created before was to sort of make this data more accessible to a wider community, and I think that's a really key output for some of these studies, because otherwise they're very challenging to analyze without making these things more accessible. So really to summarize our biological findings, we use single-cell RNA-seq to identify cell composition shifts right within these tumors, identifying, for example, the CXCL13 T cell population. We were able to identify programs that show different polarization states across, that are some of which are shared and some of them which are unique. For example, the interferon stimulating gene. We identified covariation of programs that led us to identify multicellular interaction hubs. In particular, two hubs that we studied further: this inflammatory hub and the immunity hub, one of which occurs at the luminal surface and one occurs deep within these tumors. And one thing that we didn't have a chance to characterize systematically, but seemed to be at least an anecdotal result at this juncture, is that many of these very strong hubs appear to be linked to spatial structures within these tumors, right? So we really went after two, and 2 out of 2 ended up being spatially organized. I think that it begs the question to how much does this generalize to every tumor? In terms of methodological contributions, right, I developed this consensus NMF and this covariation consensus NMF approach, right, and multiple scalable cloud-based tools for estimating the saturation and sequencing, ambient contamination, and identifying cells of poor quality to filter them out from the analysis. Really need to thank many folks who were involved, right? So this is some of the people who were instrumental in the study. I really need to single out the Cohen group, Nir, Karen, and Jonathan Chen. My postdoctoral mentor, Aviv Regev, Orit Rosenblatt-Rosen, who was also instrumental, Moshe Beaton, who is currently my host at the Weizmann, Anna Anderson from the Evergrande, and Myros Giannakis is currently my host at the Broad Institute, and also all of the funding agencies and our patients. And I'll be happy to take any questions you might have.
Right. Thank you so much, Matan, for the inter-team talk between computer science and a lot of biology here. So I think I want to check with Jenny whether we can have the authors, the audience speak directly. We have Azaf Rotem here. You want to say, you want to ask the question directly to Matan, Azaf?
How's it going, Asaf?
Yeah, I think we need to unmute Asaf.
Okay.
Yeah.
Do you do it? Do I do it?
Yes, Azaf is unmuted. Right. Hello.
Oh, hi. Yeah. Thank you very much, Matan. That was a great talk. Yeah, so I wanted to ask you about
Asaf was also instrumental in the study, by the way. So to give credit where credit is due.
Thank you. So I wanted to ask you more of maybe less usual case for these type of studies. So the normal cells, or what was slightly remote from the tissue, the malignant tissue. How are they look like in terms of any pre-malignant signals in them? Any difference between them, or maybe covariant genes that you can see something about, like what is it? It's probably not the complete normal tissue.
Yeah, I think it's a great question, and something that I have been meaning to look more deeply into. For one thing, right, there's the finding that I sort of breezed through, that they sort of present unique sets of signatures per patient, right? So to me, that tells that there is something going on at the malignant, at the normal level, right? So these are these adjacent normals, right, that might not be completely normal, either due to the presence of tumors, right, or due to something that was going on before the tumor emerged, right? So I think it's a good question that the short of it is I don't have a clear answer, but I think it's well worth looking at further.
All right. Thanks, Azaf. You satisfying with Matan's answer?
Yeah, I'm waiting to ask more questions, but I'll give a chance for other people.
Yes. So we have Imran Fanaswala. Imran, can you unmute yourself? Imran cannot talk. So the question of Imran was that it seems you have 3x more programs than clusters. Question, did you check the co-occurrences of programs and cluster? Second, is it possible that a higher resolution clustering, that means more cluster, would assemble the programs?
So I think the short answer is that yes, it's sort of, there's no obvious answer, right? So it's sort of, there's clearly, right, one could use an approach like NMF to do clustering, right? Like if one asks themselves why has graph-based approaches for clustering been so powerful for single cell analysis, I think there's a technical reason for that, as much as, I mean, they are useful and robust ways to do clustering, but they are also the ways that sort of correspond best with our TSNE and UMAP visualization. So I think that's the key reason they've become so popular, at least in my opinion. So on some level, yes, one could find some correspondence between some populations and some clusters, right? So you could always, if you were sort of to divide things sufficiently, right, they would resemble programs on some level. However, some programs are really these shared patterns, right? And while we try to create a dictionary that is as comprehensive as possible, right, by looking at the different compartments separately, I think there is some redundancy in there, right? So I think you could also always create more programs, right? So one could, it's sort of like there's a diminishing return, right? And the choice of K is always a tricky topic when you do anything in the unsupervised learning space, right? I think we sort of saw diminishing return going for more programs. But I think there's definitely a co-occurrence, right? There is certain correspondence. I think a lot of the things that, I think it even goes more deeply than that. I think a lot of the structures that biologists are very used to talk about, right? Like particular populations of cells, even, you know, like what is the difference between, you know, I hope not too many, I won't piss off too many immunologists, right? But what's the difference between a CD4 and a CD8 T cell, right? When you look in single cell data, they're actually very similar to each other, right? They sort of have some nuance, right? Some separation, right? But it's not very clear, right? So I think this question of what is a cluster as opposed to what is a continuous program is one that I think is worth further study, or I think maybe there is some contribution that we can present from the, you know, the computer science space to the biological space. But I think, first of all, we have to get all of the biologists on board with thinking about things in a continuous fashion, right?
Yes,
thinking about some things in a continuous fashion.
Yes.
We can't fight all of the fights at the same time.
So there's actually a second related questions from Yassir. I don't know if Yassir can talk, but Jenny, can you allow Jenny to Yassir to unmute herself?
Yeah. Let me.
Oh, Yassir cannot talk as well because his question I read literally as here. If I understood correctly, the loadings of the expression programs on the cell of the age matrix can be used to cluster the cells. Why do we need to use graph-based clustering in the first place? So if I understand, yes, if the
yeah. So I can try and answer. So I actually do use, right? So all of the techniques I've shown, the UMAPs these are TCs actually in this study, right? The TCs are all built on top of these NMF H loading matrices, right? So basically the underlying dimensionality reduction that I apply across the board is NMF-based and not PCA-based, right? Just to keep things consistent. And I think it also performs slightly better than PCA, at least in my hands. It's more robust, right? And it creates, I think, more interpretable structures. It also made more sense to me, right? But then the reason we then apply a graph-based approach is what I sort of mentioned before. I think it just corresponds better to these TSNE type visualizations. I don't know who in the crowd has tried to sort of look at the nearest neighbors graph overlaid on top of a TSNE or a UMAP. And I think you'll be surprised, but I don't know, maybe surprised, maybe no, but it really corresponds very well. And you could, if you look at things like force-directed layouts in nearest neighbors graph, they look startlingly similar to TSNE plots.
Oh, so essentially what you're saying is you build the care nearest neighbors and graph, and you just let that graph just pulling or pushing itself, and it.
That's what it ends up that the TSNE is very similar to this force-directed layout outputs.
Yes. Because essentially behind the TSNE or UMAP, the similar ideas was actually implemented.
I was surprised by this. I don't know how widely known this property is.
This is good. How do you choose which expression programs to keep for graph-based clustering?
So we did not pick and choose. We actually used all of the expression programs. We did do our clustering, right, in our visualizations separately, right? And the global views are most used for the discussion aspects, right? So there was a choice of K, right? But then we used everything up to that K. Although I would say that some of the right, like most of these types of approaches, I think some dimensions are likely to be, I don't want to say junk dimensions, but dimensions that are just capturing baseline activity within the cell, right? the activity of translational machinery, right? Metabolic activation, right? Things that are active within normal cells as well.
Right. So the next question from Hong Liu. I don't know if Hong Liu is still here to speak, but I read the question. So do you have your consensus NMF non-matrix factorization, non-negative matrix factorization, and the CCNMF methods available to public?
They're semi-available. I really need, like, if there is interest, please write me and we could figure out a way to share them. I mean, I'm obligated to share all of this stuff, right? And parts of it are in GitHub, but I should probably do a better job of documenting how to use it.
Exactly. Back issues. I know Matan was a C programmer, but I don't know if things changed now.
There's some C in the WeedWorks, but a lot of it is I'm embarrassed to say MATLAB.
Okay. So there's a question from Wendy Wu. How did you annotate the programs with biological functions?
Yeah. So we actually, like, we did a manual annotation, right? And that's not why not everything has a label, right? When you dig into it, I basically created certain analysis frameworks for annotating, right? So basically, I do gene set enrichment on the characteristic genes from each one of the programs, which we then look at the genes and the gene set enrichment and sort of try to find a biological label for them. I think this is very much a challenge, and I think part of it is the fact that right, the legacy knowledge, right, what we have in these databases is sometimes just not great, right? When you compare it to what we can capture today using single-cell approaches, right? A lot of the legacy knowledge in biology just doesn't hold up, right? You sort of try to use resources like MSigDB, right, or Go categories, right, and they're not great, right? We really need systematic efforts from, I think, more biologists to create right annotation sets that we could use in the single cell era.
And when did you want to say something in addition to that? I see you raise your hand.
Oh no, I just I was trying to say I'm not able to find the unmute button, but it popped up to me, just a minute ago. So
yes,
thank
good.
you.
All right, thank you. And Sinlu, you have a questions? I see you're raising the hand.
Hi, great talk and great work. So my question is about the hub. So you showed that the hub is very close at the stroma epithelial cell border. Any specific program in the stroma cells close to the hub have been observed?
Yeah, I can't say exactly off the top of my head, but definitely, one of the things that we were really hesitant in our study to make strong assertions about the stromal cells because they're not well represented. I think we were probably overly cautious because we do get, it really varies from patient to patient, where it is as much as 5% in a total of 15,000 stromal cells, which makes it a non-trivial stromal cohort still at this point. Right, we don't describe this very at great depth in the paper, but you could look at some of these programs within our companion website, right?
Okay. Thank you.
Okay, if that answers the question. I think there's further work to be done, and I have a bunch of ideas that I'm following up on how to use additional publicly available cohorts in CRC and combine them in I think a more robust way to uncover also interactions with these smaller, noisier subpopulations.
Thank you, Sin. There's another question from Rahul. A general question, given that single-cell technologies are still evolving in proteomics, what do you think is the correlation between transcriptome and proteomes? For example, you have tried a bulk proteome approach with deconvolutions.
Yeah, I've not. I think it's an interesting question. You know, first we have to get transcriptional deconvolution, right? Which is something I'm working on. I think that's the first step, right? So once we sort of can do bulk deconvolution of RNA, of bulk RNA, right, we could sort of think about the proteome. I think we'll probably need, right, further studies of single cell proteomes to do this well. I think there's a lot of work to be done on all fronts, right? But I think I've been mostly focusing on the transcriptome aspects from like just the scale at which we can characterize, right, and the comprehensiveness. But I'm, you know, like if somebody has a single cell proteomic technique that works well, I'm super excited to look at any data that they generate.
Yeah, and Rahul, you want to add anything to your questions?
No, that was a great answer. I only asked because I, do some of this laser capture micro dissection of whole cells, but I primarily work with human brain tissue, and there I see some lack of correlation between transcriptome and proteome. That being said, who knows these answers? So
yeah, and I think it's right. There's definitely some, like, there's certainly regulation that occurs after transcription, right? And it really depends who you answer, right? You ask a proteomics person and they'll tell you, oh, like, we're not capturing anything. You ask a transcriptional expert and they'll say, oh, it's at least 80% of the signal, right? Not to mention that I think even when you do proteomics, you don't study phosphorylation and glycosylation, right? So there's still a lot of work to do.
That's a great answer. Thanks.
Thanks, Rahul. And we have Imran having a questions that if you check CXCL3 plus or 13 plus cells, I wish for proliferation cycling markers. I think.
Yeah, I do not remember off the top of my head. We saw proliferation in all sorts of places, and it was actually like one of these shared signals that you see in all sorts of populations, right? So various types of, both immune and epithelial, and even some stromal population showed activity of what looked like cycling or proliferation programs, basically. We sort of see a cycling or proliferation program active within each compartment almost.
Right. All right. So I think that is all the questions we have for now. Are there any other questions? Otherwise, I would like to thank Matan so much for, extremely enjoyable talk. And Susan, by the way, by the time I thank Matan, Susan's asked a question.
We can take one more question.
Yes. So the question is, can you explain that in identification of gene expressions programs, how to get many solutions for each cell type, and how do you select one among them?
Yeah, so this is one of the things that actually is maybe a small nuance that I'm not sure I explained very well, right? So we basically uncover these dictionaries, right? These W matrices in the NMF solution, right? It's basically a set of bases vectors, right? That we uncover using NMF, right? And then we can sort of use that basis set, right? This W matrix to uncover an H matrix, a loading matrix, in any cell type we want, right? I don't know if that answers the exact question, right? So basically, we can uncover something in T cells and then look at its activity in epithelial cells if we wanted to, right? We don't really use that all that much. We sort of tend to use the T cell dictionary within T cells and epithelial dictionary within epithelial cells, right? So within each one of these cell compartments, right, we then have to choose a K, right? And we actually explored, right, different metrics for choosing the best K, and the answer is that I don't think there's a great answer, right? We used variance explained at the end. I look at other measures like silhouette, right, to basically try and really it's a heuristic, right? I think if you add more data, there might be more characteristic programs you could find. That said, I think there's a diminishing return, right? So the further on you go, the less variance you see explained in the solutions, right? So it's basically, we use the short answer is we use the heuristic that is based on finding like an elbow within the variance explained.
Yes. Yes, and we have one more questions from I see Tree is raising their hand. Can you unmute yourself, Tree, and talk to Matan? Yeah, somehow Tree was not able to unmute himself. But anyway, I think we can take this offline.
Okay.
Yeah. So Matan, thank you so much for presenting this.
Thank you for the invitation.
No, this is our great pleasure. Thank you, Diana. And I look forward to seeing you sometime very soon. Especially the situation of COVID is already ends almost. And thank you so much, everyone, for joining the talk with Matan today. All right.
Thank you. Bye-bye.
All right. Goodbye.
Thank you. Bye-bye.